Austin, are you going to
So,
be our instructor today?
yes, I will.
Oh, excellent. So this webinar is really my fault. We've had a number of so we run this multi-user facility. We have common tissue culture rooms, that multi-user tissue culture rooms, and we've had. I would say we get a report of a contamination event maybe once a month across our 108 companies. It's just not high frequency. They have escalated a little bit more recently. And one of the things that we're really trying to do is, so when you go online, you say, okay, I want a really clean tissue culture. What's the best practice? You get all this GMP stuff, which isn't relevant to us, right? We're not going to do bunny suits. We don't have changing rooms, that's the GMP stuff. And then you go and you look at, forgive me, academic labs or traditional, and it's like a mess. And I'd love to
Yeah,
find a way to be somewhere in the middle, somewhere that's
some balance there. Yeah.
yeah. And so we're really looking for you to help us both, procedurally, which is hard. Sterile technique is really the essence of avoiding contamination. But it's a little bit like when you go to doctors and you say, there's been this surgical problem. Yeah, I know about it. The guy down the hall is terrible. I myself have never had a bad surgery. I'm never the source of contamination in our tissue culture. Obviously, it's the incubator. And so it's hard for us to ever say to somebody, oh, your contamination, or that's bad sterile technique on your part. So one of the things I'm really keen to learn today are things that we can help surround them with, or alternatively, if we as a community can try to focus on teaching one thing a month. Okay, this month it's blank. So I don't want you to change your presentation, but I just want you to know why we're motivated to do this.
Yeah, absolutely. Yeah, and that makes sense to me. I think a lot of the content that you'll see in the presentation will address those points. But we can always do follow-up too. So that's really my role as a field application scientist. I'm essentially a free service from Corning for our customers that want to be trained on how to use Corning products or how to reduce contamination in their labs. So use me as needed, and we can do follow-up trainings and on just general topics like this, but specific maybe product usage as well. So if there's a specific type of workflow that your companies are trying to work through, whether it's 3D cell culture or scaling up, those are some pretty common topics as well that we do training on and presentations. So.
Great. And Bettina and Flavia and I will work together to make sure that resources that would be helpful can be made available through our shop or otherwise. So.
Great.
I'm glad you told them what you were looking for, Doug.
We have a real problem with one poor client right now that's having periodic fungal contamination. It's coming at a frequency of there may be 3 workers in a room together, all from the same company. They have their own room, and it's happening at a frequency of about once every other week for the last couple of months. We had the room sterilized, the biosafety cabinets, the incubators, and the whole room using VHP, and 2 weeks later, bang, it was back again. So we're investigating, autoclaving the pipette men, flushing bleach through the vacuum tube every day in case there's a reservoir in the tube itself. The HVAC.
Have they looked at their raw materials? Oftentimes, if it's frequent and coming up like that after doing a decontamination, there's oftentimes material source, which is a little bit harder to nail down sometimes. Yeah.
So we threw away all the media and each time, but and there's more than one cell line, and it's happening in both cell lines. But I said, hold it, when you got the cell lines, did your group aliquot them out? And could that have been the time when you got cross contamination? So let's say that the reservoir is in the pipette bin. When you then aliquoted them out, you've now introduced a contaminant to both cell lines. So sorry to
Yeah.
get us into the weeds. Why don't we
Okay.
get
No
started?
problem.
And
Let me go ahead and share my screen, get that ready.
Thank you so much, Austin and Bettina, for hosting this event with us today.
yes, thank you.
So I can, oh, Austin, while you're getting ready, I can just say, thank you for coming. My name is Bettina Bazzini-Lape, I am the MBC's local Corning account person. If any folks are on from the San Francisco site, you might be familiar with Tim Vernon, who is still with Corning, but he moved to Southern California and he's got into UCLA for grad school. So we're happy for him, sad to lose him up here, and I'm happy to support both or all sites until we backfill for him. With us today is Dr. Austin Mogan. He is one of Corning's field application scientists, very well versed in several things. Today, he will be talking to us about best practices around cell culture. Doug had asked us to do this discussion today, have this presentation today about just reducing risk of contamination at your locations. Go ahead, Austin.
All right, great. Thank you for that intro. Yeah, and good afternoon, everyone. Thanks for joining us today. So as Bettina said, you know, my role is to kind of help our customers and scientists with their cell culture workflows. So that includes everything from, you know, doing more advanced assays like 3D cell culture or scaling up cells. But really, for all of that, you know, one of the most basic tenets of doing cell culture as a practice is working in a sterile environment and not introducing contamination into your cell culture workflows. So that's what we're going to talk about today. We're going to really take a step back and get back to the basics here and talk about cell culture best practices and considerations for reducing the risk of introducing contamination into your tissue culture. So with that, let's jump into it. Really three major points here. So we'll start with just an introduction to tissue culture basics. We'll move into cell culture best practices, and then we'll end with a section on understanding and avoiding cell culture contamination. So really just a quick brief intro on Corning Life Sciences. So Corning's been around for a long time. Our life sciences presence has been around for over 100 years. And some of the first brands that we had generated focused on life science workflows were things like Pyrex glass. The Pyrex glassware was actually used to develop the first vaccine, the polio vaccine, and used to culture HeLa cells, which was the first cell line the first immortalized cell line ever utilized in a laboratory. And that was in the 1950s. Since then, we have developed a lot of tissue culture dishes and plasticware. That's really the main method that scientists now culture cells is using tissue culture treated plastic, and we'll talk about what that is in a few slides. And then moving from the 70s up until now, we've innovated in many different areas, such as scaling up cells with larger vessels, things like microcarriers, bioreactors, different specialized products for culturing cells in 3D. So your classic tissue culture is in a 2D environment where the cells are adhered to a flat surface. These days, there are many more advanced methods of culturing cells in 3 dimensions as spheres. And then other more specialized products as well for scaling up suspension cells like larger Erlenmeyer flasks and items for culturing organoids. So really a long list of history in cell culture. And so we're going to kind of take that breadth of knowledge that has been developed over many, many years and apply that to some of the basics of cell culture and reducing contamination. So really, how do we grow cells? You know, you can really break it down into these 4 primary components for your primary tissue culture work. Number 1 is you, of course, have to have cells. Number 2 is you have to have a vessel or some kind of container that the cells are being cultured in. You need to have a cell culture media, which if you think about cells in your body, they're surrounded by liquid, by blood, by lymph fluids, and this media functions like that. It provides the nutrients, the vitamins, proteins, and then a CO2 incubator is commonly used to maintain temperature as well as pH. So the CO2 is actually primarily utilized to maintain the pH of your cell culture. So what are the common cell culture vessels that are used for growing cells? These days, the most common method is utilizing polystyrene plastic, which is the universal material. And there's a few reasons for this. One, it's easy to manufacture. There's little lot-to-lot variation. You have good optical clarity, so you can observe your cells on the surface, and it's ultimately low cost. Some of the drawbacks, though, is that it's unnatural. It is a hard, non-biological surface. It's harder for some cells to attach, and the surface must be treated. But by far, it is the most commonly used method for culturing cells in a laboratory. So how do cells adhere to a tissue culture vessel? When a vessel is tissue culture treated, it's not really coated with something. It's actually a process that introduces oxygen species. So it's a chemical modification to the polystyrene backbone, that plastic chemical backbone. And the introduction of the oxygen species into the polystyrene introduces a negative charge in the form of hydroxyl groups and other oxygen groups. And that's what promotes the adherence of your cells. It's that negative charge. The cells essentially settle to the bottom of the flask, they attach and then spread out. One more advanced treatment of polystyrene plastic is Corning's cell-bind surface. So this is very similar to tissue culture treated vessels, but it has about a 60% increase in the oxygen functional groups that are introduced into the polystyrene plastic. And the reason why I mention this is because the cell bind treated surface can be ideal for difficult to adhere cell lines or cell lines that need to be grown under low or no serum conditions. And then we've also seen improved recovery from thaw using cell bind compared to your standard tissue culture treated vessels. So how are these vessels actually made? How is that treatment incorporated into the vessel? The historic method utilized the corona discharge and a conveyor belt. So the corona discharge was essentially electrical energy that utilizes the atmospheric gases to incorporate those oxygen species into the polystyrene backbone. This was developed in the 1960s. It wasn't very efficient, and so that process was modified in the 70s utilizing radio waves and O2 plasma and placing those vessels into a vacuum chamber. And that gave much more consistent tissue culture treatment on the plastic surface, and that's what is currently used today. The cell bind treated surface is very similar, but it utilizes microwaves instead of radio waves and N2O plasma to introduce that increased number of oxygen species into the polystyrene plastic backbone. So how do cells actually adhere to the surface though? Because the cells themselves are not actually directly adhering to the polystyrene plastic via that charge. They're actually adhering via either serum proteins or proteins that have been coated on that tissue culture vessel. And so that's one of the main reasons why serum FBS is often used in a tissue culture, besides the fact that it provides many of the growth factors and nutrients. But the serum proteins actually settle down to the surface of the vessel, and the integrin binding proteins that are present on the surface of the cells interact with those serum proteins, and that's what allows them to adhere to the tissue culture plastic. So as cells float down to the surface, the integrin proteins on the cells interact with the proteins that have now coated that surface, and they start to adhere and spread out until they end up flat. Now, many people ask, can I reuse my tissue culture vessels? And besides the fact that you're risking the possibility of introducing contamination into your next experiment, it's not recommended to reuse them because it's very difficult to effectively clean off the proteins that have coated the surface of that vessel. And so you're going to increase variability into your cell culture because many of those proteins that had coated the vessel in your first experiment will be left over. And when your cells go to try to adhere, they may not be the optimal proteins. They may be covered due to cell debris. So it really introduces variability into your cell culture, and that's why it's not recommended to reuse the tissue culture plates or flasks. So there's a variety of different surface coating options as well, besides just using serum to promote the adherence of your cells. Corning actually offers a long list of extracellular matrix proteins and biologically coated surfaces, from animal-derived options such as collagen, laminin, to more synthetic options that are animal component free and that are more ideal for experiments where you don't want to introduce animal components into your cell culture. This is a document that kind of has a list of many of the advanced services that we offer, and some publications associated with cell lines and cell types where those surfaces have been utilized. So there's many different considerations when you're trying to choose a cell culture vessel, and some of them are listed here. So really the first and primary is, are there certain cell biology requirements or application requirements that would push you to choose a certain cell culture vessel versus a different vessel? So that can be a cell culture vessel that is coated with a biological coating. If you have a certain cell type that, for example, needs a collagen coating, if you have a certain cell type that needs better oxygen distribution in the vessel. So the first consideration is this vessel compatible with the cells that I'm trying to culture? Second is equipment and space. So when you're working with, for example, T flasks, as you scale up and need more and more cells, you may be limited on the amount of equipment and space that you have. And so there are some vessels that, for example, have multiple layers that allow you to consolidate many of those T flasks into a single vessel. This can also save on labor. So, working with 10 T flasks versus a single vessel can decrease the amount of handling steps and labor that is associated with your cell culture workflow. There may be some investment in technology or expertise if you're utilizing a cell culture method that's a little bit more advanced, like maybe a bioreactor. And then lastly is the cell or product yield. So how many cells do you need, or how much virus do you need? And this is probably one of the more common considerations when we talk about alternative methods for doing cell culture beyond just a micro plate or dish or a T flask. Is I need more cells. What are the best methods or vessels to get me to have more cells? And so there are a variety of different options to consider for that, for scaling up adherent cell culture. At lab scale, most often you're using a microplate or 6-well plate, as well as a T-flask. You can then move into intermediate scale, such as something like a multi-flask with multiple layers, or a roller bottle, or a hyper flask. And then moving into large scale, you may consider something like a cell stack. It's a multi-layer vessel, a hyper stack, which has even more surface area per volumetric footprint. And then even beyond that is the cell cube, which pairs with a bioreactor system. And then there's a few options that aren't on this slide that are even larger scale, things like fixed bed reactors and microcarriers. The main theme here, though, is when you're talking about larger vessels, is the density of cells per surface area. So essentially what you're doing here is you're simply just scaling up the amount of surface area, the amount of centimeters squared that's available for your cells to grow. And so you often want to maintain the same seeding density, like how many cells you're putting into the vessel per surface area, per centimeter squared. And you're usually seeking the same harvest density. So how many cells are you getting out of that vessel per centimeter squared? And that's how you compare between vessel sizes. Culturing cells in suspension is a little bit different, and we're not going to spend too much time on this workflow today, but many of the considerations I'm going to talk about are the exact same. The main difference between culturing cells as being anchorage dependent versus suspension is that they don't need a surface to adhere to. You can put them in something like an Erlenmeyer flask, a spinner flask, or a bag, maintain agitation or mixing, and the cells may remain suspended and then expand. There are, certain cell lines that are amenable to being cultured in suspension, but I would say a majority are anchorage dependent and do require a surface to be adhered to for their growth. In this case, you're talking about cells per mil, or the number of cells per volume, when you're scaling up or doing your seeding density. So you typically try to keep a consistent cells per mil when you add cells into the vessel, and a consistent cells per mil when you harvest those cells and passage them. In addition to the vessels, there's a variety of other equipment and materials that are needed. These include things like vials, water baths, liquid nitrogen tanks and freezers, consumables such as tubes and pipette tips, an incubator, of course, as well as reagents. I just throw this list up here just to kind of use it as a reference. But it's important to understand everything that goes into your cell culture, and this will make sense later on when we talk about routes of contamination, because beyond just the environment and the scientist or the operator, one of the other potential routes of contamination are the equipment or materials that are being utilized in your workflow. All right, so that was the introduction to cell culture. Now we're going to talk about best practices and a little bit of a brief overview of the handling steps and the workflow itself when you're culturing cells in your laboratory. So again, we're kind of taking this back to the basics, and let's first talk about thawing cells. So the best practice for storing cells is in vapor phase liquid nitrogen. This slows down the biology and keeps the cells from dividing or producing proteins. And so this can maintain your cells for a very long time and maintain them in a state where they can continue to be used. For thawing these cells out, we usually thaw them in a water bath or warm water that has been taken into the biosafety cabinet. And to do this, you'll typically stick your vial in that water, swirl it until you see that the cells have thawed. Usually take those cells, put them into fresh media, spin them down, and the purpose of the spinning down is usually to remove the cryopreservation reagent, which often is DMSO, because it is toxic to cells over a long period of time. We spin that down, remove it, resuspend the cells in fresh media, and then they're ready to be plated into new vessel. Okay, so here we have just some basic terminology for passaging cells and working with cell culture. So the first is confluence. When you're talking about confluency, it's essentially a visual measure of how tightly the cells are growing in your vessel. So if you look on the left, we have 3 different cell lines, and you can see in the left column that those cells are still somewhat spread out from their neighbors, and so we would call this semi-confluent. The cells on the right are confluent, meaning they've reached a point where they've taken up much of the space. And the reason why this is important is because this tells us when we need to harvest the cells and passage them or split them. Because if the cells get too confluent or over confluent, it can impact the biology, and you can see a downstream impact in your cell cultures. So we passage the cells by harvesting them off the surface and then reseeding them into a new vessel. The number of times that this is done is called the passage number, and we do like to keep this below a certain number, usually between 20 and 30 passages before we throw out that cell culture. And then the time it takes for the cells to double in number is a value called doubling time. And this is essentially a measurement of how fast they are growing, and can help you dictate, when you may need to passage the cells later on. Estimating confluence is a little bit of an art, and it can be different depending on the cell line that you're working with. And so you really need to get familiar with the cells that you're working with to understand when they should be passaged before they become too confluent. So very similar explanation to what I just had. You have a low confluence to moderate to greater than 95% overconfluent. Overconfluent is when the cells are really stacking on top of each other, and you end up with a cell subpopulations of cells that have moved out of exponential growth phase and other cells that are still in that phase. One other thing to point out is many people utilize a pH indicator in their media called phenol red. And as the cells grow, they produce usually lactate, which makes the media more acidic, and it changes the color from red to a more yellow-orange. So to passage your adherent cells, the basic steps are here. So you essentially grow them to your desired confluence, aspirate out the old media. Oftentimes there's a buffer wash step. Add in your dissociation reagent, trypsin is commonly used, incubate the cells for a period of time, observe them under the microscope to confirm that they're rounding up, and then you can inactivate that solution. Depending on the dissociation reagent that you're using, inactivating it may involve just adding serum or FBS or diluting out that solution. There's some more gentle reagents that you don't necessarily have to add serum. You can just dilute it out with a larger volume of a cell culture media. Oftentimes, you're going to then spin the cells down, resuspend them in fresh media, because that dissociation reagent, if it carries over into your next culture, can inhibit the ability of the cells to reestablish and grow. Once they're in fresh media, you add that to a new flask. So during that harvest process, you're going to do a cell count. So many people, historically split their cells by just doing a ratio, meaning they'll harvest their cells and then they'll do a 1 to 5 or 1 to 10 ratio of their cells with fresh media. I strongly recommend actually doing a cell count so you can understand exactly how many cells you're going to be adding into the new vessel and have a target seeding density of cells per centimeter squared. There's a few different ways of doing cell counts. The historic way was using a hemocytometer, which is a glass slide that essentially has these boxes or grids that allows you to count the number of cells present in those grids. There are some more digital methods of doing cell counts these days. One example is shown on the screen here using the Corning cell counter, and it's essentially a hemocytometer with a digital microscope that does automated cell counting once you've added your cell sample to the glass slide. And of course, you have pipetting here, and then once you've done your cell count, you do a calculation, figure out how many cells you want per the surface area of your vessel, add it into the vessel, and then you can proceed with your downstream assays, or allowing those cells to simply expand to a larger number. Okay, and then lastly, one of the last common steps in the cell culture workflow is freezing cells. So the most commonly used cryopreservative right now is DMSO. It's a solvent, and it protects the cells from when they're frozen. So it essentially integrates into the cell membrane and protects the cells from being damaged during the freezing process. Some other cryoprotections that are often used are serum, and then there are some serum-free and DMSO-free reagents on the market as well. But really the overall workflow here is to do the cell harvesting steps that we just talked about, make sure that you have a cell count, because you typically want to freeze your cells at a specified density, and it's usually anywhere between 5 and 20 million cells per mil, or in one vial. So you would do your cell count, spin them down, resuspend the cells in your freezing media, and the rate at which the cells freeze is actually quite important. If they freeze too quickly, it can damage the cells and damage their ability to recover. If they freeze too slow, the same thing can happen. And so, what we typically target is about a degree Celsius decrease in temperature per minute. And there's a few ways to do that. One of them is using a alcohol method and placing the vials in a bath of alcohol. And the product that's often used for that is the Mr. Frosty. But there's alternative methods such as the Corning methods that do not use alcohol. They're alcohol free, and they essentially allow you to preserve your cells without adding alcohol to the container. You usually do that in a -80 degree freezer, and then transfer them a day or so later into vapor phase liquid nitrogen. That's used for long-term storage, so that low temperature -196 degrees Celsius is ideal for the long-term storage to, again, slow down the biology and make sure that you maintain the cells in the same state as which you froze them at. Okay, so the last few slides here before talking about considerations around contamination are just overall cell culture best practices. And for these details and kind of understanding of the best practices, there's not a great way to present it without just kind of going through them in a bullet point fashion. And so let's first talk about antibiotics. Antibiotics are commonly used in cell culture to help reduce the potential for contamination. They're not ideal for long-term continuous usage, and the reason for that is twofold. The first is it may hide a underlying contamination in your cell culture, whether that is a bacteria or mycoplasma, and they also can, once they hide that contamination, it can spread to additional cultures without you really observing a major impact at first if there's antibiotics in your cell culture. They can also, over a long period of time, impact the biology of your cells. And so many people eventually remove antibiotics from their cell culture, and they are still used and are good for short-term usage. So if you're working with primary cultures or you have precious samples from a biopsy, they can be used for short-term culture to establish those cell cultures. before moving on and taking the antibiotic out of the media. You should employ some controls and best practices around freezing your stocks. So it's really important to have a validated primary stock and stored in liquid nitrogen, just in case anything happens to your working stock cultures, just in case they do get contaminated, or you run into a issue where you damage the cultures by adding a reagent that you shouldn't have, or whatever. There's a long list of things that could happen to your cell culture. So it's really important to have a validated primary stock that you've tested for contaminants and that grows with certain characteristics. So you can compare your cell culture later on to that validated primary stock. This essentially removes the evolution of your culture. So you can always go back to your stock cultures. And it's important to test for common contaminants like Mycoplasma and make sure that your stock culture does not have those contaminants. And of course, just to reiterate, it's good to store these cultures at below 130 degrees Celsius to ensure that they are not drifting in terms of their genetic drift over time. When you're passaging cells, cells grow old just like we do. And so, like I said before, we want to avoid using cells that have more than, I said 20 to 30 passages earlier. That's probably the maximum. Some people prefer to keep it below 20. And then best practice also is to discard the cells that are older than 3 months in culture. When it comes to incubators, there's some considerations around incubators, particularly around the temperature, the balancing, and vibrations that can all impact your cell culture. So for temperature, you want to avoid constant opening of the doors. Watch out for a potential bad calibration. If you have an overheated incubator, that's typically associated with that calibration, as well as a underheated incubator. Changes in humidity can also impact your cell culture and cause condensation or evaporation of the media in your cell culture vessels. It's also possible that the shelves might be imbalanced, and this can impact the distribution of your cells in that vessel as well. So make sure that you have balanced and level shelves. And vibrations have also been found to be another cause of different morphology and different distribution of your cells in growth vessels. And this can be caused by other equipment surrounding the incubator. All right. So I think we've thoroughly gone over an introduction to cell culture and some of the best practices. Now what I'd like to do is talk about specific considerations around avoiding contamination and best practices to ensure that you are maintaining a sterile work environment. So some common problems that are observed in cell culture that may or may not be due to contamination, but oftentimes a contamination presents itself as one of these observations. So these include things such as low cell viability, low recovery of your cells after thaw, change in the cell morphology that you haven't seen before, cells that are not adhering properly to the tissue culture vessel, or cells that appear fine but do not respond to the stimuli as expected, or don't have the expected biology or results that you may have expected in your experiment. Some potential causes of this could be the availability of nutrients, some issues with your serum or other raw materials that are either animal derived or have some variability in them due to their source. The culture conditions themselves. So has something changed in the incubator? Is the incubator not working right? Or has there been a drop in temperature, CO2, or simply poor aseptic technique that has introduced a potential contaminant into the tissue culture vessel. So that's what we're going to spend a lot of time now talking about, is aseptic technique and some of the routes of contamination and some of the common contaminants as well that could be introduced into your cell culture. So first, before moving on to that, usually the first step in a contamination is actually identifying that you have one. And so there's a few things that you may want to look for as you're doing your experiments. And if you see these things, you may think to yourself, is it possible that I have a contamination in my cell culture? So the first is death or loss of cells. Something doesn't look right. You know, oftentimes the presentation of a contaminant or contamination is not gross, it's not obvious. So even if you see something that is slightly off from how your cells have been behaving before, it could be a potential contamination. Next is that your incubator smells funny. So a lot of bacteria and fungi have a very distinct smell to them. Or if you're getting unexplained results. So why is your culture behaving a certain way? Why are you getting results that are unusual from what you would expect? And then some more obvious things are variations in growth. So you're seeing a slower growth, or the morphology is different. So the cells look very different from what they did previously. Now, if you suspect that you have a contamination, or you've confirmed that you do have one, oftentimes it's a group effort to identify the source of that contamination. And as you'll see in the next slides, it can come from many different areas. It can come from the raw materials, it can come from the operator, it can come from the environment, but it's important that the group as a whole gets together, clearly discusses and states the problem, evaluates the possible causes, identifies the most likely cause, and then really examines and confirms the true root causes of where that contamination came from. Now, last note here is that contamination is very unlikely that you will ever completely eliminate it from your cell culture, but it can be managed and reduced both in the frequency as well as the seriousness of the contamination. So there are some common factors that can lead to contamination. So the first are cell-derived contaminants. The second is media-derived contaminants, contaminants from the environment, such as the incubator, and then lastly our aseptic technique. And we'll talk about those in more detail throughout the rest of the talk here. Now, in addition to the routes or sources of contamination, there's a long list of types of contaminants as well. And some of these may not be very familiar to you. People don't often think of toxic chemicals or other cell lines when they think about contaminants. They usually think about bacteria, yeast, or fungi. Those are the most common. But there are the term contaminant can really be anything that has been introduced into your cell culture that you don't want there. Okay, so that can be bacteria, it can be yeast, fungus, it can be chemicals, it can be viruses. We'll talk a little bit about mycoplasma because that's a very common contaminant. And then the last is other cell lines as well. And so sometimes people don't actually realize that you can cross-contaminate your cell lines, and it's often very hard to actually identify that just by looking at the cells growing in the dish. So it's important to maintain aseptic technique, and even when you're working with different cell lines, to not cross-contaminate those. So we're going to get chemical-derived contamination out of the way because it's a little bit more difficult to identify as the root cause, and we're going to spend most of the time on the biological contaminants. So chemical contaminants, there are a long list of different chemical contaminants, from metal ions to endotoxins, to extractables and leachables from plastics, to free radicals that are derived from photoactivation from components in the media, deposits left when cleaning and disinfecting your equipment. This is one of the more common ones. And then lastly is impurities in the gases, associated with your CO2 incubator. So these are a little bit more hard to identify and usually end up being determined last as the last root cause. But more commonly, it's a biological contaminant that is introduced into the into the cell culture. So bacterial contaminants are quite common. You can usually visualize them by seeing a change in the turbidity of the media or a pH change. And oftentimes you can actually detect them by looking at your cell cultures under the microscope. You're going to look for motility and size or shape, uniformity. And some of the common sources of bacterial contamination are operators, cells that have already been contaminated when you bring them into the lab, raw materials such as sera or media, unfiltered air, or the incubator itself. Fungus and yeast are also common. They can form more macroscopic structures such as clumps. And it actually is usually easier to identify them over bacteria or something else. They often show signs of budding, and they may initially form small colonies on the surface of the media. Very similar sources as bacteria, and it's usually either the operators or the environment or equipment that is introducing these into your cell culture. Viral contaminants happen less frequently. But they are one of the more difficult to detect because of their small size. You can't visualize viral contaminants, and they're also quite difficult to remove because of their small size. And oftentimes, the sources of these are contaminated cells, your raw materials, unfiltered air, or the incubator. And you usually need some more advanced tests to be able to determine if it's a viral contaminant in your cell culture. Now, I want to spend some time on Mycoplasma because many people haven't heard of Mycoplasma, but it's actually a very common issue in cell culture. So it's actually estimated that between 11 to 15% of cell cultures in the US are contaminated with Mycoplasma. It's also very difficult to detect visually because of its small size, so you cannot see it on a standard microscope. And you usually don't see any changes to the media either. So no turbidity changes, no pH changes. And the challenge with Mycoplasma is there may not be any obvious changes, but it can alter a lot of the biology of your cells. So you may see altered metabolism, altered synthesis of DNA, RNA, or proteins, chromosomal aberrations, differences in the composition of the cell membrane and morphology, as well as signal transduction. So biological impacts, but really sometimes hard to identify. And so really because of that, best practice, and a lot of labs are transitioning to this. Still, many of them do not do this, but it really is becoming more common practice. Best practice is to routinely screen your cell cultures for Mycoplasma contamination. There's a variety of different methods that can be used to do that, and many of them are listed here. But it's something that many people already have Mycoplasma contaminating in their cell cultures, and you don't realize it until you do some kind of biological study and the cells are not behaving like they're supposed to. And oftentimes that's when you identify or go down the rabbit hole and determine that it's due to Mycoplasma. So strongly recommend testing routinely for that. How do biological contaminants and
Can I chime in, Austin?
question? Yes.
This is Bettina. I just wanted to comment that I believe MBC is using Corning media in many cases, that we do routinely screen for that Mycoplasma, which just takes out one more one more thing to look for. So just wanted to state that before you move on to the next topic. Thank you.
So how do biological contaminants enter into cultures? And we've already touched on this, but the primary ways are through particulates in the air or through aerosols, through manipulation of the cell culture. So through the process of adding in your cells or working with the tissue culture vessel via transport. So if you're moving your plate from the biosafety cabinet to the incubator, during incubation in the incubator itself, if your incubator is not clean, in many incubators there are fans that essentially move the air around to maintain temperature, and sometimes that can cause air to get into your cell culture vessel if it's unscrewed or if you just have a plate top on it. But more often than not, it is technique and associated with not having the most optimal aseptic technique. And the reason for that is there's biological contaminants everywhere. So on the right are just some examples of where they are on us and the number of microorganisms that are present on different areas of our body, as well as our saliva. And sterile clothing as well after it's been sterilized, about 6 hours, and this was from one study where they sterilized a lab coat, and after 6 hours there was on average between 1 to 6 organisms per centimeter squared. So even if you're using PPE, over time you can get organisms on it that can be introduced into your cell culture. And then of course air as well. And so that's why we use a biosafety cabinet to try to limit or maintain a sterile air environment when you're working with your cell culture. So some of the common root causes or ways that contaminants are introduced can be from contaminated solutions or samples, faulty sterilization. So, autoclaving is a very common method for sterilizing things. However, if you pack out the autoclave or if you have something closed, it oftentimes does not get successfully sterilized. So for an autoclave sterilization to work properly, the steam actually has to get into all of the areas of the item that is being autoclaved. One of the advantages of buying things from suppliers like Corning is that many of our products are already sterile and you don't have to worry about sterilizing themselves and having a robust process for doing that. Some of the other sources are sharing cell culture reagents, and then lastly is bad technique during cell culture. So what is aseptic technique? Essentially, it's a set of behaviors and actions to keep contamination out of not only your cell cultures, but your reagents, the supplies that you're working with, as well as keeping the things that you're working with, if they might be dangerous, off of our cells. And so it's really difficult to kind of present practical aseptic technique theory over a webinar. It's best taught live as a hands-on training, but it's more of a mindset than individual practices. So it's more of thinking about, when I pick this up, am I going to be touching this sterile material with something that's non-sterile? So, I mean, one obvious thing is these pictures right here. You have somebody that's grabbing the stripette with their hand, which is unlikely to be sterile, right? So it's more of a group of practices and mindset of when to touch things, where to place things to maintain the sterility of your cell cultures and your reagents. So I can talk about some helpful hints though. Let's start first with the laminar flow hood. So do not rely solely on the UV lamp as the source of your sterility. It does lose its efficacy over time. Use IPA, use 70% ethanol, wipe down your area before and after work and between cell lines. Limit traffic to the area around the hood while you're working. Wipe off your bottles with IPA before placing them inside the hood. Avoid talking while you're working, or wear a mask because aerosols can be introduced into the hood. And don't use a laminar flow hood for long-term storage of things. The last thing I'll say here is that the laminar flow hood maintains sterility, but only if you use it properly. And so in many of these hoods, there's flow or cycling of air through a HEPA filter. And if you block any of the air vents, or you block the ability of the air to flow over your work area, you're limiting the potential of the hood to maintain sterility of your working environment. Some other helpful hints here we have around transporting cells. So keep the distance as short as possible. Use sealed vessels. So put your plates or your dishes in a separate container. Avoid spills and liquid bridges. So for example, if you have a microplate and you accidentally tip it too much, you can form a liquid bridge of the liquid coming out of the wells and interacting with the top of the plate. And transport dishes and microplates in boxes or Ziploc bags to maintain the clean environment when you're going from the biosafety cabinet to the incubator. For personal protection or PPE, it's really important to wear clean or disposable lab coats with fitted sleeves. So if you have your sleeve that's open, you could absolutely be introducing something into your sterile work environment from the exposed skin. Ask yourself, how frequently are your lab coats cleaned? Do you have dedicated lab coats for cell culture versus bacterial work? You absolutely should. You should also have dedicated incubators for cell culture versus bacterial work. Are you using cuffed sleeves for cell culture? So making sure that you don't have exposed skin inside the biosafety cabinet. Wash your hands, use gloves, spray your gloves with ethanol. Hair can be a dust magnet, so if you have long hair, put your hair up. Don't bake or brew before culturing. So this is something that may not be obvious, but if you're working with yeast at home, you can bring that into your cell culture lab. Don't have plants in the lab either, because they're essentially harbors of contaminants. And make sure that you decontaminate all of your culture waste properly and do not store it for long term in your lab close to where you're actually doing your cell culture. Some other considerations are around reducing airborne contaminants. So place sticky mats at the room entrance, have a schedule for cleaning the floors. And then the last few points here are around your caps. So historically, people have kept their caps slightly unscrewed to allow gas exchange. That can be a source of contamination as well. The ideal situation is to utilize a vent cap that has a non-wettable PVDF membrane to allow gas exchange to occur without having to keep that cap unscrewed. So some final considerations around the equipment and materials to maintain a sterile work environment. Make sure you change the water in your water bath as well as the water in your incubators for humidity frequently. They can be areas that harbor contaminants. Regularly disinfect both your water bath and your incubators. For the media and solutions, dispense them in single-use quantities. Sterile filter your media, use separate media bottles for every cell line. Don't share bottles, don't double pipette, and never work with more than one cell line at one time. Pipetting is probably one of the most significant areas of contamination. Use a pipette tip typically only one time. Keep in the wrapper until use. Change the tip or pipette if it touches anything or if it comes out of the hood, and use filtered pipette tips with your pipetters. One of the last slides here is around reducing contamination utilizing a larger vessel strategy. So one of the ways that we introduce contamination is if we're working with many vessels at one time. So for example, working with 10 tea flasks, you're more likely to contaminate one of those or many of those versus if you're working with a single vessel that gets you the same amount of cells or surface area as 10 tea flasks. And so there's a variety of different vessels that can be used to consolidate the number of vessels that you're working with, and that can reduce your risk of contamination. That's this point here, more surface area. Second is introducing closed system solutions into your workflow. So these are bottles, bags, caps, tubing assemblies that allow you to transfer liquid without pipetting or pouring inside the biosafety cabinet. So you can make sterile connections and do all of your liquid transfer, even outside of the biosafety cabinet using these closed system solutions. And please interact with myself or or Bettina, if you want to talk about some of the solutions here that can be used for closed system transfer. So I'm at time here. There was a lot to cover, but the risk for cell culture contamination can be minimized with aseptic technique. Try to take a proactive approach, because not only is it aseptic technique, but you can reduce your risk by maintaining cleanliness in your lab. Incorporate routine screening with kits or stains in your workflow. And there's also multiple options for designing a more optimized process to reduce the risk of contamination, such as not reusing your vessels, increasing the vessel surface area and not number of vessels, move from an open to a closed system. And we'd be happy to talk to you more about some of those closed system capabilities. We have some more information on our website. Feel free to reference it here. And I know I'm at time here. Happy to take some questions. I can stay on a little bit longer, but thank you for your attention, and I'll be happy to take some of your questions now.
Thank you so much, Austin and Bettina. I think it was a very informative session. And I think Doug has a question here, right, Doug?
Yeah. So are those closed systems suitable for 3D cultures?
It depends on the workflow. So are you doing 3D cultures at small scale still, or are you looking at scaling up 3D cultures?
That's a good question. It's not me. I don't ever do any science anymore. But let me bring that up. Actually, Bettina, we may want to talk to Resistance Bio about that.
Okay.
And then a question, a sort of general question, which is, if I were to inoculate one well of a 12-well plate, and I would use super serial technique to inoculate it, how often would I get contamination across the other 11 wells? In other words, most liquid-borne bacteria or fungus are going to have a very hard time getting aerosolized sufficiently to transfer within a closed plate, aren't they?
Oh, that's true. Yeah. So within a closed plate, you're talking about transfer between wells, correct?
Right.
Yeah.
Right.
So
I guess what I'm thinking about, forgive me, it's going to sound defensive on my part, but if you think about how many bacteria fungus could get from a water bath in an incubator inside a plate that has a lid on it, it's going to have, I mean, that's a really torturous path, because unless you're aerosolizing.
Mm-hmm. Sure. So the likelihood of aerosolizing is low. I would say, in my experience, most of the time it is associated with contact. So it's associated with you touch a part of your microplate or flask. However, the best practices around the incubators and the water baths is, if you have those clean, the number of contaminants that are on the materials that you're bringing into the biosafety cabinet, or the materials that you're carrying with you, is reduced, right? So you're absolutely right.
I'm not arguing the incubator should be dirty. I'm just thinking, God, because you walk across the lab. One of the things I really liked that you showed was the 25 centimeter square plates for transporting to the incubator. Do you leave those inside those large square plates during the incubation, or do you take them out when you get there?
Yeah, so you can leave them in those. You just want to make sure that they're not covered, because you do need the access of your tissue culture flask to the CO2. So you can leave them in those dishes for transport and then place them in the incubator. I would actually transport them covered and then take off that cover once it goes into the incubator.
And then when you do the opposite, when you come back the other way. Yep.
Exactly.
Okay, very cool. Wonderful session. Thank you so much, Austin. Bettina, thank you so much for arranging this. Much improved. I have to leave, but everyone else can pepper. Now that I've had my shot at Austin, everyone else can take their turn. Thank you so much.
All right, Doug. Thank you. Take care.
Any
Go ahead, Bettina.
Any other questions that might be specific to your application? Open to discussing those.
Yeah, we can go beyond just, talking about contamination as well. If you have any specific workflow, like anything to do with cell culture, you're having any issues, we can kind of open it up to that as well. Or we can even have a follow-up one-on-one call. So feel free, if there's any outstanding issues with your experiments right now, we can certainly talk about those too.
Well, if we don't have any questions, the session was also recorded. We can share the webinar with you guys. And thank you once again, Austin and Bettina. It was amazing. And we are looking forward for the next session.
Yeah,
Thanks, Flavia.
Thank you for reaching out and letting us know what might be relevant topics for the group. I know it could be, certain topics could be relevant to some and not to others. So thank you for letting us know what would be useful right now.
Of course, our pleasure. Thank you so much, everyone.
Great.
Thank you. Bye-bye.
Take care. Thank you. Bye.
Bye.